صباح الخير جميعا، يعطيكم العافية. حنبلش بانترو صغيرة بس عن الكلاب قبل ما نبلش بالانترودكشن للايفنت ومع اور سبيكر. نحن نادي بايوس أمادا هو نادي البيولوجي الأول بكلية العلوم الفرع الأول الحدث. اتأسس عام 2019. كان هدفه الأساسي إنه نحن نوجه الطلاب ونساعدهم بمسيرتهم الدراسية، وهذا الشيء عم نعمله من خلال الندوات التوجيهية اللي بنعملها كل سنة لكل السنين. منتابعهم وما بعدها منحاول نساعد الطلاب بمسيرتهم المهنية، وإذا حبوا يكفوا كبي اتش دي عبر مختلف الإيفنتات بمسيرتهم المهنية، عبر إنه نأمن جوب فيكنسيز، نساعدهم ليحضروا للانترفيوز، CV writing, motivation letters, LinkedIn profile, etc. وكبي اتش دي هيدا الشيء عملناه من خلال هيدي الايفنت اللي هي Research Without Boundaries 2، اللي هي بتخليهم يجتمعوا مع مختلف طلاب اللي خلصوا PhD واللي هن UL students بالأساس، ليشوفوا research وين عم يروح، شو الاشياء اللي عم تنعمل scientifically، ويقدروا يستفيدوا من تجربتهم لحتى يكفوا إذا هن حابين. باختصار هيدا الايفنت هتكون عم تساعدكم لتعرفوا على scientific part، بنفس الوقت تقدروا تعرفوا كيف بدكم تكفوا your pathway. وأكيد بهمنا نعمل شوي جو فن مع تعليمي، وهذا الشيء بنعمله بمهرجان العلوم الكبير اللي بنعمله كل سنة بالعلوم بالحدث. أهلا وسهلا فيكم جميعا. هيدي الإيفنت بلشت من السبت الماضي وكفت مع كل السبيكرز بمختلف بلدان العالم. اليوم آخر نهار لنا وحيكون عنا سبيشال بارت حنقدمه نحن كبايوس، هو virtual lab tour لحتى تشوفوا كيف الأجواء اللاب وهيك. إن شاء الله اليوم بعد الظهر. حنبلش مع our second speaker for today سالم. بليز تفضل وأهلا وسهلا فيك وميرسي إنك معنا.
Hello everyone, thank you for this opportunity. So I'm Salem Al Siblani, a Master 2 student at the University of Grenoble Alpes, studying Physiology, Epigenetics, Differentiation and Cancer. And today I'll be presenting for you my Master 2 thesis entitled Toward Dissecting by Proteomics, the links between deregulated metabolism and rewired transcription in Huntington's disease. This project has been supervised by Dr. Delphine Fliguer and the CAA Grenoble and the EDIT team in collaboration with the LNCA lab in Strasbourg and the BrainC lab in Paris. So to start with, what is Huntington's disease? Briefly, Huntington's disease is a neurodegenerative disease that attacks mainly, but not only, a specific region of the brain that's called the striatum, as you can see in this figure. And it leads to the neurodegeneration in that region. This neurodegeneration is accompanied with movement and cognitive disorders. Among the early hallmarks of Huntington disease, the patients are mainly characterized by a deregulated metabolism. We are mainly talking about the glucose metabolism that's being disrupted, along with dysregulated epigenetics and gene transcription at very important neuronal identity genes. Our goal in this project is that we're trying to understand more what are the links between these two and Huntington disease, and understanding more the link will open the way to new relevant targets of treatments to treat this dysregulated gene expression. But what is the link between both of them? Glucose through glycolysis will produce metabolites. These metabolites are considered the substrates for histone post-translation modifications, denoted as PTMs, and these histone post-translation modification can control the gene expression. So we can see a direct link between the glucose metabolism and the control of the gene expression. As an example, the glucose through glycolysis will give rise to acetyl-CoA, but that's not the only source of acetyl-CoA. And then acetyl-CoA in the nucleus, under the effect of specific enzyme, will deposit its acetyl group in a process known as histone acetylation on the histones, and this is usually accompanied with an active gene expression. We have a vast variety of histone PTMs that can take place For example, we also have the histone trimethylation, which can be either associated with gene expression or gene repression, depending on the site that's being modified. These modifications take place on the histones, and a histone is a protein that's wrapped around the DNA, and the modification that occur on these histones will decide whether the gene will be expressed or repressed. The histone is an octamer of proteins, and it's composed of H2A, H2B, H3, and H4. And in this study, we are going to focus on the histone modifications on the lysine residues of the H3 and H4. The general and final goal of our study is to determine how the perturbations, how the disruptions in these glucose metabolisms are delivered into histone post-translational modifications in the context of Huntington disease. As a hypothesis that we have in mind, we have proposed that the Huntington disease patients will have different kinetics of histone PTMs. And by kinetics, I mean turnover rates and the speed of the addition and removal of these histone acetylations that are specifically derived from the glucose metabolism, as you can see in this figure to the right. So the black curve corresponds to specific turnover rate for histone acetylation, and the proposed hypothesis says that this curve in red for the Huntington disease patient gonna have different kinetics compared to the white type. To be able to validate this hypothesis, we are going to quantify the level of specifically glucose-derived histone PTMs and compare it between white type and Huntington disease mice. And this will be done by injecting the mice with heavy glucose and then following the incorporation of the heavy atoms from the heavy glucose into different histone PTMs. So this was the general goal of all the study. But in my internship, we're starting from scratch, and we need to establish the basics of this study. For this, we need to establish the protocol of histone extraction from nuclei, and we also need to establish the pipeline for the data analysis. For these reasons, we are going to work only on the white type mice and on their striatum, on their hemisphere. And here's what we did. Our collaborators in Strasbourg, they worked on the white type mice only. They deprived these mice from glucose for 1 day. After 1 day, they applied intraperitoneal injection of heavy glucose. They waited for 30 minutes. They applied another dose of subcutaneous injection. And then they isolated the hemisphere of these white type mice after 30 minutes, 60 minutes, and 120 minutes of the latter subcutaneous injection. We in Grenoble extracted the histones from the hemisphere of these white type mice, and then we run the histones on gel to be sure that we have really extracted the histones, and we can estimate that we have approximately extracted 200 micrograms of histones from each hemisphere. These histones undergo some cleavage and some processing to make them suitable for the proteomic analysis by liquid chromatography mass spectrometry, which will identify and quantify all of these peptides and thus their modifications. This data was analyzed with Mascot, Skyline, PCOR, and R software. And here's more about what's going on. So the heavy glucose that we used was the uniformly labeled C13 glucose, denoted as U13C glucose. This is a glucose in which all of its 6 carbons have been substituted with a heavy carbon. This heavy carbon contains an additional neutron, and it's denoted as C13. And then we're going to follow the incorporation of this C13, this heavy carbon, from the heavy glucose into different histone post-translation modifications, and we expect to see a specific mass increments profile for each histone PTM. For instance, we expect to see the incorporation of 3 heavy carbons in each lactyl-CoA, and lactyl-CoA is the substrate for histone lactylation, so we expect to see the mass increment of approximately +3 dalton for each heavy histone lactylation. And histone lactylation has been very recently discovered in 2019. Also, we expect to see the incorporation of two heavy carbons in each acetyl-CoA, and acetyl-CoA is the substrate for histone acetylation. So we expect to see the mass increment of +2 daltons for each histone acetylation that's being labeled. And at the end, we expect to see the incorporation of one heavy carbon in each SAM, and SAM is the substrate for histone methylation, and thus seeing the mass increment of +1 for each methylated group. But this pathway from glucose to SAM is a bit long, and we may not be able to see it, to see the incorporation within our time points. So these mass shifts will be detected with liquid chromatography mass spectrometry, and it will allow us to differentiate between the heavy histone post-translation modifications that are derived from the glucose that we have added, this heavy glucose, and the light histone post-translation modifications that are either already existing or they have been derived from other sources other than the glucose. And here are the results. We had a big list of results, but I decided just to show you 4 of them. And if you focus on the first chart, you see this one corresponds to the peptide, which is the N-terminus of H4, containing 4 acetyl groups on lysines number 5, 8, 12, and 16. And these colored bars correspond to the number of heavy acetylations among these 4 acetylations. For instance, this orange bar right here, it corresponds to the heavy version of this peptide, where it has 1 heavy acetyl group and 3 light acetylations group. And as you can see, we have a maximum incorporation of C13 after 30 minutes of the heavy glucose injection for this peptide. And we also see a similar trend in the rest of peptides where we have a maximum incorporation after 30 minutes of the glucose injection, which decreases with time. But interestingly, if you look at the H3K27 acetylation, the last graph, you see that we have a big difference between 30 minutes of injection and after 60 minutes of injection compared to the rest of the peptides. And this tells us more about the fastest or faster turnover rate compared to the rest of the peptides. And this can be due to that the H3K27 acetylation is well known to regulate the gene expression of genes that stimulate to cellular stress, for example, like the heat shock proteins, which requires fast response from the cell. So as a summary, we were able to detect the incorporation of C13 into histone acetylation after the injection of heavy glucose. However, we weren't able to detect the incorporation of this heavy C13 into histone methylation, nor into histone lactylation upon the addition of our heavy glucose, and that there may be some site-specific kinetics of histone acetylation exchange, just like the case we saw in the H3K27 acetylation. And in the next step, we're going to test if you can get histones from the nuclei of the striatum specifically, and not from the whole hemisphere. And after that, we're going to apply a single injection of the uniform labeled C13 glucose and compare it between white type and Huntington disease with a smaller difference in time points to have a better idea and better understanding of the kinetics that's going for these histone PTMs. And on the long term, we are considering the analysis of the histone post-translation modifications in astrocytes and neurons separately. At the end, I'd like to thank all for listening, and special thanks to Delphine Flieger and Hassan Hijazi, who is the third-year PhD student, and for our collaborators. Thank you. And I'm open if you have any questions.
ميرسي كتير سالم على الانفورميشن برزنتيشن الحلوة، وإذا حدا عنده أي سؤال أكيد فيكن عم يسأل.